Welcome to FDA Grand Rounds. Today's speaker is Dr. Jayaleka Amarasinghe. She's a microbiologist at the Winchester Engineering and Analytical Center, also known as WEAC, within the Office of Specialty Laboratories and Enforcement Support in the Office of the Chief Scientist here at FDA. Since joining the agency in 2014, she has led pioneering research on characterization of biofilms associated with medical device surfaces, driving efforts to develop innovative antimicrobial-free strategies aimed at enhancing medical device safety. Dr. Amarasinghe earned her undergrad degree in biology from Bay Path University in Longmeadow, Massachusetts, and her doctorate in oral biology from the University at Buffalo, State University of New York. She also completed a postdoc in infectious diseases at the New York State Department of Health. Her work has been crucial in advancing novel approaches to anti-biofilm technologies for medical devices. Welcome, Dr. Amarasinghe, and the floor is yours.
Thank you for that kind introduction, Rakesh. Thank you for joining, and welcome everyone to the presentation. Today, I will be presenting on anti-biofilm technologies for enhancing the safety of medical device surfaces. So this is the standard disclaimer. The views expressed during this presentation are my own and do not necessarily represent the official position of the FDA. And any mention of commercial products, trade names, manufacturers is for clarification only, and it is not intended as FDA approval, endorsement of recommendation. So before diving into the presentation, I'd like to briefly share where I am based. I worked at the Winchester Engineering and Analytical Center, or we call it WEAC, just north of Boston. And WEAC is the FDA's lead regulatory testing lab for medical devices and the radiological health program. We are proudly led by Brian Baker, our amazing director, and a true champion of scientific innovation. Here's a quick overview of what I will be covering today. I will start with the public health impact of biofilms, then move into the challenges of controlling them using traditional methods. From there, we will explore emerging anti-biofilm strategies, and I will highlight some of the WEAC's recent work in this space, including the electrical stimulation and photothermal ablation as anti-biofilm technologies. I will wrap up with key findings and where we are heading next. So to begin, let's take a moment to define what a biofilm is. These slides illustrate the classic model introduced by Kostetter and colleagues. A biofilm is a highly organized community of microorganisms encased in a self-produced matrix called extracellular polymeric substance, or EPS. Once planktonic or free-floating bacteria find a surface with enough nutrients and a moisture, they start to colonize and then form structured cooperative communities like this. So as this biofilm matures, the bacteria secretes EPS, a sticky protective matrix made of polysaccharides, proteins, lipids, and also extracellular DNA. This matrix is not just structural, it's also defensive. It shields the microbes from antibiotics and environmental stresses, and also host immune responses and in disinfectants. All components of the EPS contributes to this biofilm resilience against the antimicrobials. Finally, in the last stage, the part of the biofilm disperse, releasing bacteria to colonize new surface and restart the life cycle. So understanding this cycle and especially the role of the EPS matrix is critical for developing strategies that can disrupt or prevent biofilm formation. So, biofilm-related infections are a major public health concern, especially in healthcare-associated infections, or we call them HAIs. These infections contribute to about 100,000 deaths annually in the US alone, and over 60% of the HAIs are linked to medical devices contaminated with biofilms. So biofilms are extremely difficult to treat because they resist antimicrobials and often leads to device failures requiring surgical replacement. This is not only impact patient outcome, but also contribute to a massive economic burden estimated at about 28 to $45 billion annually. So this pie chart shows the global economic impact of medical device biofilms with central venous catheters and prosthetic joint infections according to accounting for the largest share. So now that we have seen the scale of the problem, it's important to recognize the range of devices affected by the biofilms. So implantable devices like prosthetic joints, dental implants, breast implants, and also central venous catheters have been associated with serious biofilm infections due to long-term implantation and limited immune access. And also reusable and reprocessed devices like endoscopes and heater cooler devices or HCDs have also been linked to outbreaks when biofilm persists despite standard cleaning protocols. So this slide highlights how widespread the contamination is and also underscores this need for strong anti-biofilm strategies across both implantable and reusable device categories. So now we have seen how widespread the biofilm infections are, but treating them is even more difficult. Biofilms can be up to 1,000 times more resistant to antibiotics than their planktonic counterparts. This is due to the coordinated set of defense mechanisms that protects the microbial community within the biofilm. These include the EPS matrix that blocks antibiotic penetration and also bacterial communication system called quorum sensing system that allows bacteria to detect their population density and activate group defenses. And within a biofilm, there are persister cells and that hides inside the biofilm and then enters into a dormant state to evade treatments. In addition, there are multiple other mechanisms such as efflux pumps that actively expel antibiotics. And also within the biofilm, there are microenvironments containing low oxygen and low pH pockets that reduce the drug effectiveness. So together, these barriers leads to poor antibiotics penetration, incomplete eradication, and frequent relapses. That is why there is a strong need for physical and antimicrobial-free strategies that can more effectively disrupt biofilms. So given biofilms resistant to antibiotics, alternative strategies are essential. So this slide highlights emerging approaches integrated into medical devices uh, to prevent or disrupt biofilms through non-traditional mechanisms. This includes physical methods like electrical fields, vibration, photothermal light, therapies to break down biofilm structure, and also chemical approaches such as pH modulation and localized antimicrobial release using polymer drug conjugates, as well as biological strategies like antimicrobial peptides and quorum sensing inhibitors that disrupt microbial communications are also being explored currently in the field. So smart coatings and surface modifications also been exposed to help prevent bacterial adhesions and enable on-demand antimicrobial release. Carrier-based systems like these micelles and like lipozymes also enhance delivery into dense microbial layers. So together, these innovations represent a multidisciplinary approach to improving the safety and effectiveness of medical devices. So many of these strategies we have discussed are now being translated into real-world application. But it's important to distinguish between what's experimentally promising and what's clinically validated. For example, silver nano-coated endotracheal tubes and electroactive wound dressings are already FDA cleared for general antimicrobial use or wound healing, though they do not carry any biofilm-specific claims. Photothermal therapy, photothermal coatings using gold nanorods and magnetically responsive wound dressings are under active development with encouraging lab scale results, but have not yet reached clinical approval. On the biological side, antimicrobial peptides like LL37 are being incorporated into catheters and coatings, while quorum sensing inhibitors and CRISPR-based hydrogels are showing promise in preclinical studies but remain unapproved for clinical use. Even bacteriophage therapies, which have gained renewed interest for resistant infections, are still largely investigational and not yet FDA approved for device-related biofilms. So while the pipeline is full of exciting possibilities, most of these technologies still require robust clinical validation and regulatory review before they can be marketed with biofilm-specific claims. So as we just seen, numerous emerging technologies are being explored to combat biofilms. However, many of these approaches are still in early stages and have not yet been fully validated or approved by FDA for biofilm-specific applications. So at WEAC, we are contributing to this space by evaluating the safety and effectiveness of select promising technologies within a regulatory science framework. Our group is specifically focused on medical devices, assessing innovative anti-biofilm strategies and that could enhance device safety. Currently, we are leading two key projects. One, investigating the use of electrical stimulation to disrupt nontuberculous mycobacterial biofilms on stainless steel surface, and the other explores nano-functionalized titanium surfaces against bacterial biofilms. These efforts involve not only the evaluation of existing methodologies, but also the development of robust, reproducible protocols tailored to slow-growing, clinically relevant bacterial species like NTMs or non-tuberculous mycobacteria, which have been linked to outbreaks and persistent device-associated infections. So let's begin with our first project I'd like to share with you. And this work focuses on addressing non-tuberculous mycobacterial biofilms using electrical stimulation as a non-traditional approach on stainless steel medical device surface. So to give some background, non-tuberculous mycobacteria or NTMs are widely present in the environment and include over 200 known species. So they are typically classified as either rapid growing, which forms colonies within 3 to 7 days, or slow growing, which can take 14 days or more to show a colony. So NTMs are known as opportunistic pathogens and especially notorious for forming biofilms on medical devices. These biofilms make them remarkably tough to eliminate. In fact, the biofilm forming NTMs can be up to 1,000 times more resistant to chlorine compared to E. coli biofilms. So NTM infections have been also linked to a wide range of medical devices, including both implantable devices like prosthetic joints, catheters, and also reusable ones like endoscopes and heater cooler devices. In rare cases, they have been associated with contact lens solutions also. So these infections are extremely difficult to treat due to both the resilient biofilm structure and their high resistance to antibiotics and disinfectants. So one of the most alarming examples of a biofilm-related NTM infection is the Mycobacterium chimera outbreak linked to heater cooler devices that happened recently. These devices are used during open heart surgeries. Although these heater cooler devices don't come into direct contact with the patients, contaminated internal water tanks colonized during manufacturing allow biofilm-laden water to aerosolize and expose patient during surgery. So the biofilm persisted despite standard cleaning protocols, leading to global exposure, difficult to treat infections, and in some cases, fatal outcomes. So the outbreak triggered FDA safety alerts, device recalls, and also international health warnings. So during the global M. chimera outbreak, one of the major challenges was lack of high-resolution visual evidence showing how NTM biofilms actually form on medical device surfaces. So WEAC was one of the first laboratories to address this gap, capturing high-fidelity images of M. chimera biofilms on clinically relevant materials such as stainless steel and titanium surfaces using scanning electron microscope and also 3D laser scanning microscope. So what you're seeing here is a time course comparison of biofilm development on stainless steel. So I want you to know that M. chimera are slow growing species. So it would take about 14 days to even see a colony. So we were surprised to see that within a 1 day of inoculation, we can see these M. chimera cells are attached to stainless steel. And then after a week on the left image over here, the right image over here, after just 1 week, a dense mature biofilm has formed, clearly covered by an extra polymeric-like substance. So this image captured provide critical visual evidence that even slow-growing NTM species like Mycobacterium chimera can quickly establish into robust biofilms on medical device surfaces, reinforcing the urgent need for better prevention and cleaning strategies in reusable medical devices. So now that we have visualized how robust and rapidly NTM biofilms can form on metal surfaces, the next question is how can we effectively disrupt them, especially given their resistance to conventional antimicrobials. That brings us to the objective of this project, to test whether electrical stimulation can help eradicate NTM biofilms from stainless steel medical device surfaces. On the left, you see a schematic of electrochemicals principle. Applying electrical stimulation to metal surface drive a Faradic reaction, such as oxygen and water reduction that generate hydroxide ions and also reactive oxygen species, or ROS. And these species can locally damage the biofilm structure. The graph on the right shows a polarization curve. The open circuit potential is or OCP, is the natural resting voltage of a metal in solution. So there is no current flows. To trigger biofilm disruptive chemistry, we apply voltage beyond this OCP. So this drives the electrochemical reaction needed to generate reactive oxygen species and disrupt the biofilms. So this gives us a non-antibiotic surface targeted strategy for weakening and potentially eradicating NTM biofilms directly at device interface. So now that we have seen how electrical stimulation can trigger key surface reactions, the next question is, how does that actually affect biofilms? This slide summarizes the proposed mechanisms currently in the literature. Electrical stimulation can work through both direct and indirectly to damage the bacterial membrane and inhibit growth. And indirectly, it can alter the environment and by changing pH, generating reactive oxygen species, and also enhancing drug penetration. Together, these effects help weaken the biofilm and make the bacteria more vulnerable to antimicrobials. So this approach actually show a real promise. So we wanted to use this technology to on these slow growing, hard to eliminate non-tuberculous mycobacterial biofilms. So now we have introduced the concept of using electrical stimulation to disrupt NTM biofilms. Let's take a closer look at how the experiment was conducted. We began by inoculating the stainless steel coupons with a standardized culture of Mycobacterium chimera strain, which is a slow-growing NTM species. And then biofilms were grown for either 1 day or a 1 week in 7H9 medium at 30 degrees shaking. After incubation, the coupons were gently washed in PPS and placed into this custom-built agar chamber designed for 3-electrode potentiostatic stimulation. To ensure the uniform conductivity, PBS was added to each electrode well surrounding the coupon. When we then applied 2 types of cathodic electrical stimulation. Cathodic voltage stimulation of minus 1.8 volts versus the reference for 1 hour, or a cathodic current stimulation of minus 1 amp centimeter square current density for 5 to 30 minutes to 40 minutes was applied. And then following this stimulation, we evaluate the biofilm viability by measuring the colony-forming units from both coupon-associated bacteria as well as planktonic bacteria from the solutions. And then we have also performed live dead staining to visualize the viability of these biofilms in using fluorescent microscope. So this approach allows us to assess how well electrical stimulation reduce viable biofilms both on the surface and also in the surrounding solution. So let's take a look at our initial results, starting with the early stage NTM biofilms, which is 1 day old Mycobacterium chimera biofilms. Based on the previously published literature, we began our testing by using minus 1.8 volts versus reference electrode, a cathodic voltage that has shown effectively against Staphylococcus aureus biofilm. So we wanted to use the same voltage stimulation in our NTM studies to see what kind of effect we will see on the biofilm viability. So we have applied this voltage for an 1 hour to 1 day old mycobacterium chimera biofilm on stainless steel coupons. And it's important to note that the mycobacterium biofilms are typically require up to 40 days to form mature colonies. So this model reflects a very early stage of a biofilm for M. chimera. So after stimulation, we observe about 1.5 log reduction in viable biofilm-associated bacteria. This is equivalent to about approximately 96 reduction of the viability compared to the untreated control. While this is a measurable effect, it falls short of the 3 log reduction threshold generally considered by the FDA for intermediate level disinfection for mycobacteria. So this suggests that early stage NTM biofilms show some susceptibility to cathodic stimulation, voltage stimulation, but that minus 1.8 voltage alone is not sufficient for full eradication under this condition, highlighting the need for stronger, longer or combined strategies in the future work. Since cathodic voltage alone was not sufficient to fully eradicate early NTM biofilms, we next tested whether applying cathodic current could enhance efficacy, targeting both biofilm-associated and free-floating or planktonic mycobacterium chimera cells. So here we have applied a current density of minus 1 amp for various duration, 5 minutes, 30 minutes, and 40 minutes. And then did this same study as previously and look at biofilm viability compared to the untreated control. One of the striking finding was that high sensitivity of planktonic NTM cells to electrical current. With just 5 minutes of stimulation, we observed significant reduction in planktonic viability. By 30 minutes, we were able to completely eliminate these planktonic bacteria by 30 minutes of treatment with minus 1 amp current density stimulation compared to the untreated control. Even more compelling, we found that by applying minus 1 amp current density for 40 minutes was sufficient to completely eliminate 1 day old biofilm, results that far exceed the performance seen with voltage alone. And an increased amount of cathodic charge was delivered throughout the stimulation period, as you can see in figure C. So these results highlights the cathodic current as a promising non-antibiotic strategy for disrupting both early stage biofilms and also planktonic NTM population. Following these strong results we observed with the early stage biofilms, we next tested whether cathodic current stimulation could also disrupt more mature NTM biofilms. So we repeated the experiment using 1 week old M. chimera biofilms and applied this minus 1 amp current density for 40 minutes. And targeting both coupon-associated biofilms and free-floating planktonic cells. As with the earlier experiments, we saw significant reduction in planktonic cell population, cell viability, compared to the growth control. However, when we examine the biofilm-associated cells on the stainless steel coupons, the effect was more limited. We observe about only 2 log reduction in viable cells in this 1 week old chimera biofilm treatment. So we think that this may be due to that NTM biofilms have already begun to produce thick extracellular polymeric substance or matrix, which act as a protective barrier and limits the effectiveness of the electrical stimulation. This finding suggests that biofilm maturity significantly contributes to treatment resistance, reinforcing the need for early intervention in any electrochemical biofilm control strategy. So to further confirm our finding, we use fluorescence microscopy to visualize biofilms after electrical stimulation, applying live dead staining to the stainless steel coupon after the stimulation. In this method, we are using two fluorescence dyes, cytone 9 and propydium iodide. So cytone 9 penetrates all bacteria and stain them green. Propydium iodide only enters bacteria with damaged membrane and stain them red. When used together, this dual stain approach allows us to distinguish live from dead cells in the biofilm. On this slide, you see the image of three conditions. An untreated growth control is on the first panel over here on the top. Middle panel show a 40 minutes ethanol treated control as we wanted to test the chemical treatment compared to the electrical stimulation. And the third panel in the bottom show a 1-day-old biofilm subjected to -1 amp current density of treatment for 40 minutes. The merge image over here show both stains overlaid, allowing us to visually access the proportion of live versus dead cells within the biofilm matrix. In the growth control and ethanol treated control, you can still observe substantial green fluorescence, indicating many viable cells remained, especially for the growth control, and we see some viable cells remain in the ethanol or chemical treated biofilm control. And then in contrast, the electrical treated sample shown an almost loss of green signal, demonstrating a significant reduction in live biofilm. This visual evidence strongly supports our colony count data and demonstrates that electrical stimulation can achieve biofilm disruption level not attained by ethanol alone, at least in the early stage of M. chimera biofilms. Next, we have done the same experiment, same staining with 1-day-old biofilms subjected to electrical stimulation. You can see a mix of green and red signals, especially in the merge panel, indicating increased cell death in this electrical stimulated treated panel over here. But it did not completely eliminate all viable cells, but it has eliminated a significant portion of cells compared to the growth control. So compared to the untreated control, there is a clear reduction in biofilm viability in the electrical stimulated sample. So this suggests that the mature biofilms are more resistant and may require long exposure, longer intensity, or combination of strategies to fully eradicate. And it also highlights the challenge of treating established nontuberculous mycobacterial biofilms and reinforces the importance of early interventions. So in summary, this study helps expand our understanding of how effective electrical stimulation can be for biofilm control, especially at early stages. While mature M. chimera biofilm remain hard to eliminate, these results support further exploration of electrochemical strategies to improve safety on reusable medical devices. So we are currently exploring how electrical stimulation can boost antimicrobial effects and the electrochemical reactions involved in this NTM biofilm removal, as well as if there are potential corrosion impacts on the stainless steel. So let's talk about the project number 2. Here we are developing titanium surfaces coated with gold nanorods to target Staphylococcus aureus biofilms using photothermal laser ablation, while also assessing the biocompatibility of these functionalized surfaces using mammalian cell lines. In this project, we are developing gold nanorod-coated titanium surfaces to combat bacterial biofilms on medical devices. As some of you know, gold nanorods are promising coating materials as they can generate heat by absorbing light, and we can fine-tune this property to rapidly kill bacteria on surface without damaging the surrounding tissues. These gold nanorods absorb near-infrared or NIR light through a process called localized surface plasmon resonance, where conduction electrons oscillates collectively and then relax, releasing energy as localized heat. So we use this NIR light because it penetrates tissue effectively and is minimally absorbed by surrounding biological components, allowing safe and targeted heating of the coated surfaces to rapidly kill bacteria without damaging the nearby tissues. Our goal is to fabricate and characterize these surfaces and evaluate their biocompatibility and antimicrobial performance, addressing the current gaps in assessing gold-based antimicrobial coatings for medical devices. So this project, we had 3 aims. First, we have fabricated and confirmed gold nanorod coatings on titanium surfaces using 2 different fabrication methods. Fabrication methods with 2 different coating agents. And then second, we tested these coated surfaces for their safety on mammalian cell lines. Third, we evaluated their ability to generate heat and reduce Staphylococcus aureus under NIR light. To achieve stable gold nanorod coatings on titanium surface, we examine 2 silane-based surface modification strategies, APTES and MPTMS, each enabling distinct chemical interactions. First, we have oxidized the titanium to expose hydroxyl group for silane attachment. And then APTES APTES is 3-aminopropyl trimethoxysilane. I'm going to call it APTES. And MPTMS is 3-becaptopropyl trimethoxysilane. And throughout the presentation, we will call it MPTMS. So APTES introduce positively charged amine group, which enables electrostatic binding with citrate cap gold nanorods, whereas MPTMS introduce thiol groups that form strong, more stable, covalent-like gold sulfur bonds, resulting in gold attachment, more stable attachment. So this comparison allows us to evaluate how silane chemistry influence coating efficiency, stability, and also photothermal biofilm killing performance, helping to identify the most effective strategy for achieving robust surface functionalization with gold for medical devices. So next, we have done various characterization, particularly physiochemical characterization and biocompatibility testing, as well as photothermal activity analysis on these titanium surfaces functionalized with gold nanorods. So to assess the wettability, we measured the contact angles of these functionalized surfaces by placing water droplets on each surface. APTA treated surface showed lower contact angles, indicating they are more hydrophilic and more wettable. And in contrast, MPTMS treated surfaces were more hydrophobic with higher contact angles, showing less wettable. However, as we will see in the next slides, surface wettability alone does not determine the nanorod attachments. The strength of the chemical interaction is more important and critical factor. So here I'm showing data for characterization of titanium surfaces coated with these gold nanorods from both schemes by scanning electron microscope and elemental energy dispersive X-ray spectroscopy, or EDX, for understanding the elemental map. So you can see the first panel is the scanning electron microscope data, whereas the bottom panel shows the EDS elemental maps. So you can see that the gold nanorods are successfully attached on both APTES and MPTMS coated surface successfully. However, when you compare these two A and B images, you can see MPTMS treated titanium surface contain more uniformal and dense gold rod coatings compared to the APTES treated titanium. The green signal in the EDS map on the bottom confirms the presence and the distribution of gold with especially effective coverage on MPTMS treated titanium. So this data confirm that gold coatings on titanium surface both visually and chemical. Also next, to confirm what elements were actually present on the surface, we used a technology called X-ray photo electron spectroscopy, or EPS. This is a surface sensitive technique that detects elements within just a few nanometers of the surface. Both APTES and MPTES coated samples showed the presence of gold, but the MPTMS treated surface produced a noticeably stronger gold signal, indicating more efficient and stable nanorod attachment. So this complements our earlier SCM and EDS results, which visually we saw denser coverage with MPTMS, and chemically both EDS and XPS confirmed that more gold is actually bound to the surface. So the cross multiple methods, we are seeing a consistent pattern. MPTMS enables stronger and more uniformal GNR or gold nanorod attachment on titanium. So after confirming the presence and distribution of the gold nanorods, we assessed the biocompatibility of the gold coated titanium surfaces using a direct contact test following ISO 10993-5 guidelines. This is an international standard, outlines procedures to evaluate in vitro cytotoxicity of materials or medical device materials, helping determine whether these materials causes any toxic effects when in direct contact with mammalian cells, which is a crucial step in preclinical safety assessment for medical devices. In our setup, gold nanorod-coated titanium samples were placed directly onto a layer of mammalian cells and incubated for 24 hours, as shown in this image. To quantify the cell viability, we perform an MTS assay, which this assay measures the metabolic activity of cells. Viable cells reduce the MTS reagent, which is a tetrasodium-based compound, into a purple colored formazan product, and the absorbance intensity of this color directly correlates with the viability number of metabolically active cells. So following this direct contact setup, we assess the biocompatibility across 3 key mammalian cell types: macrophages, fibroblasts, and osteoblasts, to represent immune response, connective tissue response, and also bone response. As shown in the graph, cell viability remained high in comparable to both control titanium as well as control alone or cell culture alone for both APTES and MPTMS coated surface with gold nanorods, indicating we don't really observe a significant cytotoxicity. As expected, the positive control, we are using a polyethylene film with 0.1% PUA CDAC, which is a cytotoxic material, showed a marked reduction in cell death, confirming the assay sensitivity. So next, to assess the potential leachables from the functionalized surfaces, we perform an extract test based on the same standard, ISO 10993-5. Here, we are using titanium samples coated with gold nanorods, incubated in cell culture medium for up to 21 days, allowing any soluble compounds to leach into the medium. Then these extracts were then applied into human osteoblast or fibroblast for 24 hours, and cell viability was measured using the same MTS assay described in the previous slide. So here are the data for the extract test. Over 21 day period, extracts from both coating methods, both the GNR functionalized titanium surfaces did not reduce viability of the osteoblast or fibroblast cells. So this indicates there's minimal cytotoxicity leachables available compared to the controls, and it sustained the biocompatibility. We also look into cytokine secretion by macrophage cells next, as a way to assess inflammatory response to these gold nanorod-coated titanium surfaces. This was measured by ELISA, focusing on IL-6, a marker of inflammation, and TGF-beta, which is associated with tissue repair and immune regulation. The results show that no significant changes in cytokines levels in response to these coated surfaces, suggesting that the gold nanorod coatings did not trigger an inflammatory response. In contrast, we have also used a positive control, which is a lipopolysaccharide or LPS. It's produced a strong cytokine response, confirming that the assay was functioning as expected. So this finding indicates that the GNR-coated titanium surfaces appears biocompatible, and at least in terms of macrophage activation. Next, we wanted to test whether these gold-coated titanium surfaces could generate heat when exposed to light, a property known as photothermal activity. So here in this figure, we expose each sample to 808 nanometer near infrared or NIR laser for 20 minutes and used a thermal camera to monitor the temperature change over time. As shown here, the titanium coated with gold nanorods via MPTMS exhibit more consistent and higher uh, temperature rise, about 15 degrees after 20 minutes, compared to the APTS coated surfaces, which was kind of not that consistent, and it's kind of got plateaued and went down after 20 minutes. So this suggests more effective photothermal conversion, likely due to the denser and more stable gold nanorod attachment on the MPTMS coated treated surface. So we are not only seeing better coating efficiency, but also improved functional performance here. After confirming the photothermal heating potential of the gold nanorod-coated surfaces, we next ask whether this heat could be used to reduce bacterial biofilms. As you can see here, Staphylococcus aureus biofilms were grown for 24 hours on gold nanorod-coated titanium, then exposed to NIR light as previously shown. And titanium surfaces treated with gold nanorods via MPTMS treatment showed over a 1 log reduction in viable bacteria after 20 minutes of irradiation, which is significantly greater than what we saw for biofilm reduction in APTES treated titanium surfaces. So this demonstrates the enhanced photothermal anti-biofilm effects of MPTMS-based coating. So to summarize this project, we used two chemical methods, amine-terminated silane, APTES, and thiol-terminated silane, MPTMS, to functionalize titanium surfaces with gold nanorods. MPTMS led to greater gold nanorod deposition than the APTES. Both coatings were biocompatible, but only the titanium surfaces coated with gold using MPTMS showed effective photothermal biofilm reduction after NIR exposure. So ongoing work is focused on optimizing this approach more for broader biofilm application. And in closing, finding from both projects contributes to a growing understanding of how surface-based strategies may help address persistent biofilm challenges on medical devices. NTM and Staphylococcus biofilms remain difficult to manage, but our results suggest that electrical stimulation and laser-activated gold-coated titanium surfaces may help reduce this biofilm at early stages, emphasizing the value of early intervention. Timing was critical as mature biofilms are significantly harder to treat due to their complex EPA structure. So our findings support the need for a performance-based evaluation framework and clear regulatory pathway for emerging anti-biofilm technologies. And moving forward, we aim to refine these strategies under more realistic contamination scenarios and explore their potential alignment with FDA's goal for reprocessing and reusable device safety as well. So here is a summary of the protocols and method used throughout both projects in case anyone would like to access the detailed procedures or replicate the work. With that, I like to acknowledge many individuals and groups who supported this work. Special thanks to the research fellows and interns and WEAC management, as well as colleagues from OCS, CDRH, and CBER. And also we like to thank University at Buffalo for providing the initial electrical stimulation protocol, and our collaborator, Dr. Jerome Batuta from Cambridge for his technical input on electrochemical setup. And this research was supported by grants from the Chief Scientist, Office of Chief Scientist. Thank you so much.